Imagine this, an infectious disease, really devastating to the brain. But here's the wild part. It's caused by just a protein.
Yeah, just protein, no DNA, no RNA, absolutely nothing with genes.
It sounds completely impossible, right?
Yeah.
Like it breaks a fundamental rule of biology.
Totally. I mean, everything we understood or thought we understood about how life works, how things are inherited, how infections spread, it all came down to nucleic acids, you know, DNA and RNA.
Exactly. And yet this really revolutionary idea that a protein alone could be infectious, well, it was formally proposed in a huge paper in Science back in 1982.
And it wasn't just like a minor finding. This concept directly challenged the core dogma of molecular biology. Think about that. Questioning the absolute foundation.
It's huge.
Yeah.
And this idea, the research behind it, it eventually led to a Nobel Prize. Fundamentally changed how we see a whole class of diseases. Okay, so let's really get into this deep dive, unpack that crucial 1982 paper.
Okay, so before this paper, for decades really, scientists were scratching their heads over these weird, slow, degenerative diseases, things affecting the nervous system and animals and even people. Scrapie in sheep and goats was maybe the most famous one.
Right, Scrapie. And it got that name because the poor animals would literally scrape their wool off. Just this intense, awful itching.
Mm-hmm. But that was just one part of it. You also had tremors, loss of coordination, basically a total breakdown of brain function, and it was always fatal.
So what made Scrapie and, diseases like it such a massive puzzle for scientists?
Well, it was how they behaved. They were definitely transmissible, no doubt about it.
Right. You could take brain tissue from a sick sheep, process it somehow, inject it into a healthy one.
And boom. Eventually, after this really long incubation period, we're talking months, sometimes years, the healthy animal got sick too.
So clearly infectious.
Yeah.
But here's the kicker, right? This is where everyone got stumped.
Exactly. The infectious agent just laughed off treatments that would normally destroy viruses or bacteria.
I mean, they threw the kitchen sink at it, didn't they? Heavy radiation, harsh chemicals designed to shred DNA and RNA.
Yeah, methods that would reliably kill any known virus. Totally ineffective against whatever was causing Scrapie. This was a huge problem.
If it spreads like an infection but resists things that target genes, what is it?
Well, the main idea floating around before 82 was it had to be some kind of really weird, slow virus.
Like a virus that hid its genetic material somehow.
Right.
Or maybe replicated in some bizarre way we didn't understand.
Pretty much. Anything to make it fit the existing model, the infection equals nucleic acid rule.
But then the biochemistry started hinting at something else.
Right. As people got better at trying to purify this mystery agent, the clues started pointing towards protein. The infectious activity seemed to stick with the protein fractions.
Which must have sounded completely nuts at the time. Borderline heresy, right?
Yeah.
Against the central dogma.
Absolutely. The central dogma was clear. Information, replication that requires nucleic acids, period.
And that's exactly where Stanley Prusiner's work comes in. He was looking at that biochemical evidence, controversial as it was.
Yeah, and his core question, the one driving that 1982 science paper, was incredibly bold. Basically, could the Scrapie agent really be just protein?
Not just another option, but a direct challenge to everything they thought they knew.
Right. So this deep dive is really about how they tested that radical idea. The experimental design in the paper is, well, pretty clever.
How do they approach it?
Their main strategy was purification and selective destruction. They tried to purify the agent, then hit it with treatments known to destroy specific types of molecules, proteins, nucleic acids, fats, sugars, and see what killed the infectivity.
And they measured infectivity using animal tests.
Exactly. Biological assays. They'd make dilutions of their sample, inject them into animals, often hamsters. There were quicker models in sheep, and see how much agent it took to cause the disease. If a treatment reduced infectivity, it meant that treatment was hitting the agent itself.
Okay, so what pointed towards protein?
Well, the evidence was really strong. They did these controlled experiments using proteases.
Enzymes that specifically chop up proteins, like molecular scissors.
Precisely. And the results, pretty dramatic. Treating the agent with proteases significantly reduced its ability to cause infection.
So destroy the protein, destroy the infectivity. Makes sense.
Yeah, the more they hit it with proteases, the less infectious it became. That was a huge piece of the puzzle.
But the crucial part was comparing that to treatments for other things, especially nucleic acids, right? To tackle that slow virus idea.
Absolutely critical. They used, I think, 5 different methods specifically designed to mess up RNA and DNA.
Like what sort of methods?
Oh, things like hydroxylamine, which chemically alters nucleic bases. They use nucleases enzymes that chew up DNA and RNA specifically. Also, things like alkali treatment, though they did know alkali can hit proteins too, but they had other more specific nucleic acid treatments as well.
High doses of UV light, too, wasn't it? That usually damages viral genes.
Right, UV irradiation known to inactivate viruses. And the results from all 5 of these were just striking.
They didn't work?
They completely failed to inactivate the Scrapie agent, consistently, across 5 different methods designed to destroy genetic material.
Wow. So treatments that kill proteins reduce infectivity, but treatments that kill nucleic acids don't.
Exactly. That was the revolutionary finding. It strongly suggested that whatever this agent was, it didn't depend on DNA or RNA for its infectivity like viruses do.
Okay, so if it's not a virus, what is it? Did they manage to isolate it?
They worked incredibly hard on purifying it from infected animal brains. And it wasn't easy. The agent was there in tiny amounts, and it was apparently quite sticky, probably hydrophobic.
It's hard to separate from everything else.
Very. They used a multi-step process, things like differential centrifugation, spinning samples at high speeds to separate things by size and density, and also antibody affinity chromatography, using antibodies to grab onto specific molecules.
Sounds incredibly laborious.
Oh, it was. The paper mentions needing something like 2,000 hamsters for the experiments. Just imagine the scale.
Good grief. But did it pay off? Did they purify it?
They achieved a massive enrichment. They managed to concentrate the infectivity by about 100,000 times compared to the starting brain tissue.
100,000-fold. That's huge.
Yeah.
And what did they find in that concentrated stuff?
Critically, that highly concentrated infectivity consistently tracked with a specific protein fraction. When they analyzed this purified material, they kept seeing a protein band around 27 to 30 kilodaltons in size.
A specific protein size showing up where the infection was most concentrated.
Exactly. A kilodalton, or kDa, is just a unit of protein mass. So find this 2730 kiloday protein, and you find the concentrated infectivity. This was what they called their semi-purification stage. Strong biochemical evidence linking the agent directly to this protein.
Did they look at its physical properties too?
They did, and it wasn't like a nice uniform virus particle. It showed what they called heterogeneity in size.
Meaning it came in different sizes.
Yeah, which they thought might be because it clumps together or sticks to cell membranes, that sticky hydrophobic nature again. But based on how it behaved in filtration and sedimentation tests?
Tests that sort of measure size and density.
Right. They estimated the smallest infectious unit could be tiny, maybe 50,000 molecular weight or even less. Much smaller than the smallest known viruses. Another point of difference.
Okay, so let's recap the evidence. Sensitive to protein killers. Resistant to nucleic acid killers, purifies along with a specific protein, and has weird physical properties unlike viruses.
You got it. Based on all that, Prusiner and his team concluded they were looking at something totally new. It wasn't a virus, wasn't bacteria, wasn't a plasmid, not even a viroid, those little infectious RNA bits.
It needed a new category, a new name.
And that's what they proposed right there in the 1982 paper, the term prion.
Prion, derived from, what was it?
Proteinaceous infectious particle.
Prion. And they defined it how?
As a small proteinaceous infectious particle resistant to inactivation by most procedures that modify nucleic acids, clear, concise, and revolutionary.
But it still leaves that massive question hanging, doesn't it? How on earth does it work? How could a protein replicate or cause disease without genes?
Absolutely. The 82 paper laid out the compelling evidence that it was protein-based, but the how? Well, that mechanism was still a puzzle they were working on. That understanding came later, building directly on this groundbreaking work.
Still, the impact of just proposing it based on that evidence, it must have been enormous.
Oh, immense. The prion hypothesis wasn't just, another finding. It was a genuine paradigm shift in biology. It just completely overturned the bedrock belief that you needed nucleic acids for biological information transfer and infection.
It introduced a whole new way infection could happen, not through genes, but through protein shape.
Exactly. Protein conformation, the specific 3D way a protein folds. The idea that emerged later, built on this, was that a misfolded abnormal version of a normal protein could act like a template.
Forcing the normal ones to misfold, too. Like a chain reaction.
Precisely, a chain reaction of misfolding. Even now, knowing it's true, it still sounds like something out of science fiction, doesn't it?
It really does. And technically, the paper was important, too.
Yes. Beyond the concept, they developed that quantitative way to link infectivity levels to the amount of protein. That gave everyone else a target, a method to follow, which was crucial for eventually purifying the protein fully and finding the gene for its normal counterpart.
And the implications went way beyond just sheep scrapie, right?
Absolutely. This new understanding opened the door to explaining other really baffling degenerative brain diseases. The 82 paper itself focused on the Scrapie agent, but their reference list hinted at things like Creutzfeldt-Jakob disease, CJD, and Kuru in humans. Maybe they work the same way.
Which turned out to be the case for many of them.
Yeah, and think about the practical side too. Biosafety, sterilization.
Right. If standard methods don't kill it.
Exactly. Procedures that worked fine for viruses and bacteria, like normal autoclaving, weren't enough for these incredibly tough proteins. That led to totally new guidelines for cleaning surgical tools, for instance.
And then, tragically, mad cow disease. BSE.
Yes, understanding prions became absolutely critical for figuring out bovine spongiform encephalopathy in cattle and its terrifying link to variant CJD in people who ate contaminated beef.
The idea of a misfolded protein-jumping species. Horrifying.
It really is. So you see how this one really bold idea stemming from painstaking work on a sheep disease ended up having these huge widespread impacts on human health, animal health, food safety, everything.
It's incredible. But you mentioned it was bold. This wasn't easy research, was it? Going against the grain like that.
Not at all. Proposing a protein-only agent, that flew directly in the face of decades of established science. The source notes actually say Prusiner was risking his career pursuing this.
Wow. So it wasn't just smarts, it was serious guts.
And perseverance. Imagine those tough purification steps, needing thousands of animals, facing failures. The notes mention continuously pushing forward in the face of repeated experimental failures. Gajamion.
Yeah.
That takes incredible dedication. Believing the data when it tells you something everyone else thinks is impossible.
Definitely speaks to real scientific grit.
For sure. Now, of course, like any landmark paper, it wasn't the absolute final answer on day one.
Right, there were still questions.
Yeah, the purification wasn't perfectly down to a single protein band yet. Some critics still wondered, could there be tiny bits of hidden nucleic acid? Or maybe lipids involved?
Those details got ironed out later, finding the gene for the normal protein and all that.
Exactly. The picture got much clearer over time. But that core evidence in the 1982 paper, it was strong enough. Compelling enough to completely shift the conversation and point firmly at protein being the essential infectious component. It was paradigm-shattering, really. And despite the initial skepticism, it ultimately won Prusiner the Nobel Prize in '97.
So, looking back at this deep dive, what's the main thing to take away?
I think it's just a fantastic example of how science progresses. One research team, driven by a really bold hypothesis, backed up by rigorous experiments, can completely overturn what we thought we knew for sure.
And why does that matter for us, listening today?
Well, it's not just some old story from biology class. It shows that even the most fundamental assumptions in science, things considered absolute rules, can be challenged and should be if the evidence points that way.
And when they are challenged? And maybe overturned.
It doesn't mean science failed. It means it's working. It opens up totally new areas to explore, leading to breakthroughs we couldn't have predicted, with real impacts on our lives, our health, our safety, just understanding how life works at a deeper level.
So that 1982 paper gave us prions, infectious proteins, operating by rules nobody thought were possible.
Yeah, the whole idea that just changing a protein's shape can make it infectious, make it act like a template to corrupt other proteins. Even now, it's still kind of mind-bending, isn't it?
It absolutely is, which kind of leaves you wondering.
Right. If protein shape alone can transmit information and cause disease in this completely unexpected way, what else might be out there? What other weird biological processes using mechanisms we haven't even imagined yet might be happening right now, just waiting for someone to notice?